DECRA in my DECRA application, I've got some contingencies for fieldwork, so maybe I'll be more confidently calling myself an anthropologist after that. Because definitely getting it. Think positive. Oops, let's see how this changed slides. So I'm going to talk about the first part of the talk. I'm just going to be talking about diversity and linguistic diversity in New Guinea, or in the Papua sphere is what we're calling it, which is an area of influence slightly more wider than New Guinea itself. And then in the second half of the talk, I'm going to talk a bit about the cross-section of genetics and linguistics and how those might tell us stuff about the process of diversification. So the Papua sphere, as we're calling it, doesn't really have a clear boundary. From a linguistic point of view, we have Papua languages going as far east, is this way, right? East is the Solomon Islands and Bougainville, into New Ireland, New Britain, and then as far west as East Timor down here, and Halmahera in the north. And that's kind of the extent of linguistic lines, but we know that there's Papuan influence beyond that, with evidence of cultural practices in Vanuatu, as well as genetic evidence that Papuan people have been moving around these areas. So we keep an open mind or a fuzzy line when we're talking about the Papua sphere. But needless to say, it's an incredibly linguistically diverse area. The island makes up around 1% of the global landmass, has about 0.2% of the population, but has around 20% of all the world's languages. It's important to note as I go through this that the Papuan, or the Papua sphere is, from a linguistic point of view at least, is a negative category. And that if we compare it to something like Austronesian, this is a single language family where we know all the languages are related to each other, coming from an expansion of people from Taiwan. When we talk about Papuan languages, we're talking about basically non-Austronesian languages. So languages within that Papuan kind of category, we have about 40 different language families, the largest of which being Trans-New Guinea, which is the green one you can see there. And in this map, if it's not the Austronesian color, then, or it's not Australia, which is another division we draw, we would consider it a Papuan language. And so it's fairly obvious on this map that there's a lot of diversity, linguistic diversity there. But this is just one type of diversity. And so I want to draw this distinction between diversity and disparity. When we look, and I'm going to show how I look at Papuan languages and this dimension of disparity. So disparity is this idea that comes from biology, that we might count the number of species in any particular place. But we might also want to look at diversity across another dimension, which might be like morphological diversity. So a quick biological example would be, this is a drawing from Darwin's identification of 18 different species of finch in the Galapagos Islands. It's just 4 of them, obviously. And although there's 18 species, if you look at the morphological diversity of these birds, they're all fairly similar, particularly if you're comparing the top 2 against each other or the bottom 2 against each other. We could take 18 species from another place, say New Zealand, which is where I'm from. We might have a kea, which is a flightless bird. Kiwi is the flightless bird, and the kea is the mountain parrot. Might have the takahe, which you often find in swamps, and up to 18. And in both senses, would have 18 species, so there'd be the same level of diversity. But this is where the dimension of disparity really becomes clear, because we wouldn't say that they have the same level of morphological diversity, for example. And we can make the same comparison with language. If we took 5 European languages, they're all going to be pretty similar because they're all from one language family, Indo-European, but they're also have similar sociological environments. The big nation, languages are linked to a big nation, they're heavily ingrained in the education system and so on. If we took 5 languages from New Guinea, chances are they're going to be from different language families. And we often infer that different language families means more time apart from each other, more divergence, and so we would expect a high level of disparity. So I'm going to kind of test this idea of disparity relating to linguistic time depth in a sense, using these 2 databases that I worked on in my career, the first being Kimbank, which Stacey mentioned in the introduction, and you can visit at this website, and please do. And this was the majority of the work for my PhD, was collecting kinship terminologies and understanding their diversity. So there's around 1,200 languages, or 1,229 according to this website, SNP. And basically we have a genealogical grid, which we're going to use to compare across languages, and we apply kin terms to each part of the grid. And GramBank is another project I worked on, which was led by Hedvig Skjergaard, who did her PhD at ANU. And that contains around 2,400 languages and 197 different variables of grammatical diversity, which are largely thought to be independent. And so what I'm going to do is compare in all the languages that we have data on to understand their structural diversity in the terms of the kinship terminology. So we're not really looking at the word forms, just the structure of the system. And in the GramBank analysis, we look at principal components of the data set. But it'd probably be easier just to talk about an actual result. So this is the kinship terminology diversity with a focus on Papuan languages. So there's 3 different subsets of kinship terminology here. Parents and parent siblings, which is parents and parent siblings, but we also make distinctions on relative age, so elder or younger, on gender as well, and also if it's a male or female speaking. So you're talking about someone who's the same or opposite gender to you. Siblings is a 8-category system, so elder brother, elder sister, younger sister, and whether a man or woman speaking, and then the same again for siblings and cousins, but including people that are further away. Then each column we have is a different subset of languages. So global is the total diversity that we see in the kinbank set. Then we have NTNG, which is nuclear Trans-New Guinea. Trans-New Guinea is like a really big family, but some areas are contentious, but there is this kind of core element that's mostly agreed on, which is what this is showing. Then we have other families of other Papuan families. So this is a combination of Timor Oriental, which is down in East Timor, Yam, which is southern New Guinea, Sepik, which is Sepik in the north, and North Mahara family as well. Then we're comparing to 3 other language families: Austronesian, obviously in the Pacific, Pama-Yungan is the largest Australian language family, and Indo-European, mostly in Europe. So this particular sample is mostly in Europe, although it does spread it to Asia, the whole family. And so what we do when we're asking about disparity in kinship terminology is to say, for any particular family, how much of this area here makes up of the total diversity. So just as a percentage. And what we see when we look at Papuan, we treat Papuan as a category, Papuan diversity is making up 65 to 70% of all observed diversity, which is pretty high, particularly when we compare it to Indo-European, which is very not diverse or not disparate. If you know anything about Indo-European kinship, that's kind of not that surprising. Most of the languages there have like a nuclear family system, so uncle and auntie word, mother and father word, brother and sister word. Maybe they gender cousin, maybe they don't. French and English, for example. What was kind of surprising about this, though, was the extent of diversity in Austronesian, which we didn't really expect, to be honest. So we're making this link between disparity and time depth. So the more time languages have to diverge, the more disparity we should see. But compared to Austronesian is a relatively young language family, we would have thought. So because this paper was mostly about Papuan languages, we haven't really thoroughly investigated it. But 2 kind of proposals for why that might be the case is, firstly, the Austronesian covers a large area where there are lots of other language families. So it could be that Austronesian has a really high level of contact as it's moved down through Asia, Southeast Asia, across New Guinea. So it's 3 kind of big areas. And then obviously Pacific as well, but largely unmeasured. Or it could be that we just track, in the case of kinship terminology, we're just tracking this link to social organization. And Austronesian has a particularly broad, may have a particularly broad collection of social organizations, ranging from cousin marriage systems to more nuclear systems, and so on. But that's something we need to investigate more, because it was quite surprising to find that extent diversity in Austronesian. The last thing I want to mention is in Pama-Yungan, because people always ask about it. Why is there one point up here in Pama-Yungan? This is Kala Lagua Ya, which is a Torres Strait Island language, and that in fact quite close to New Guinea. So we think in this case, this is probably example of some kind of contact between Papuan languages and this Australian language. And if we kind of imagine drawing a dot over, it kind of fits in some of the New Guinea language families. But so in this case, the Papuan language diversity is really, really high, which is what we did expect. But it's also comparable to the language families where we didn't expect there to be so much disparity. This is the same idea, but for grammatical diversity. And this paper, Papuan languages, wasn't the focus, but I'm just kind of pulling that out to talk about. And here we use geographical regions rather than language families. There's three geographical regions for New Guinea: the North Coast of New Guinea, South Coast of New Guinea, or South New Guinea, and the interior. And again, we get some parts of what we would have thought, that New Guinea is quite a geographically constrained area, but still has really high grammatical diversity, particularly if you look at the North Coast and the South Coast, which is the two areas where we see a lot of language families. And we compare it somewhere like the African Savannah, huge geographic area, similar levels of linguistic diversity. But once again, Oceania has crazy levels of diversity, which we didn't expect. So it's kind of a thorn on the side of this disparity relationship to linguistic time depth. Interior New Guinea is comparatively less diverse. I think you can see there's much fewer languages there. Partly this is because there's just less documentation in the interior than there is in the other two areas. So we may see that there's more diversity there than we expect, but also the interior New Guinea is where Trans-New Guinea is thought to have started from. So there's a lot of Trans-New Guinea language families there. So we might expect this disparity there as well. So that was like my quick spiel on like linguistic diversity in New Guinea, or the Papua sphere. So we know there's a really high number of languages, high number of language families, and also we're seeing that kinship systems are more disparate than we would find in Europe and Australia, and the grammar is also very disparate. So diversity is kind of, we have diversity across these two dimensions, I guess my point. But as much as we're interested in diversity, I think most people are interested in how it arises rather than just showing that it exists. So I'm going to just talk about, I'm just going to take my shoes off because I've taken my shoelaces have come in and I don't want to so I'm going to talk about a few theories about why there's so many languages and why there's so much linguistic diversity in New Guinea. And most of these ideas come from my discussions with Nick Evans, who is nominally the head of ECDI. So these are align a lot with his ideas, but I mostly agree, I think. So the first one is state centralization. So, or in the case of New Guinea, absence of state centralization. The idea here being that we can make some assumption that historically small speech communities would have been the norm. You might have had groups of like 1,000 people if they were hunter-gatherers, maybe 40,000 people if agriculture was involved. And they would have maintained their own linguistic community. But when you start to get really large states, they standardize communication by having some kind of institution that you have to engage with to get any benefit. And so communities that were otherwise linguistically diverse kind of lose that linguistic diversity as a result of engaging with the state. And there's not really any evidence of large states existing in New Guinea prior to colonial arrival. And even in the case of colonial influence, there's a lot of movement in terms of the state. So you have a German colony, you have a British colony, Netherlands colony, Australia comes in at some point before Papua New Guinea comes in the 1970s. And still Indonesia very prominent in West Papua. So the kind of, even though there has been some states influence in New Guinea, hasn't been that stable anyway. And so this is an interesting hypothesis because I think it's more reflective of what we see happening now. We see increasing use of lingua francas like Tok Pisin being used alongside an increasing rate of language engagement in the country. But I'm not sure how much we can use this to explain. This is kind of like an absence of evidence hypothesis in terms of if we're thinking about linguistic diversity prior to colonial contact. And it also doesn't really explain why there's so many languages in New Guinea, but there's comparatively less languages in Australia, which have two areas of similar time depth. The next hypothesis I'm going to talk about was isolation building diversification. This is a very simple evolutionary idea, I guess, where the longer two groups are apart, the more they diverge, similar to what I was talking about when it came to disparity. And in New Guinea, people apply this theory by saying it's very inhospitable terrain, very steep in some areas, and also a very plentiful environment. So the kind of desires for people to move between groups is perhaps low. And that drives this linguistic diversification. And that kind of logically makes sense. I don't think it really fits in that well with how people are experiencing New Guinea at the moment, where we know there's a lot of connection between groups. People are very multilingual, people connected through quite complex marriage systems. And in particular, where networks, which is a picture here, in southern New Guinea, it's actually very flat anyway, and we still have a lot of linguistic diversity. So these guys, networks in an area where a language is spoken called Nen, and it's about 20 kilometers away is another language called Edi, and they're both different language families. People get between them all the time. So I'm not sure how much isolation from a geographic point of view is going to explain diversity, but we should consider it, I guess. Second to last is time depth. People have just been there for a really long time, so maybe that's why there's so many languages. Human occupation started around 60,000 years ago in Passmore, which was the supercontinent of Australia and New Guinea, with people getting to the east coast of New Guinea up to around 49,000 years ago. And if we take some basic rules of thumbs around how quickly we think languages diverge, some people have proposed 1,000 years for a language to diverge. If one language just arrived in New Guinea 60,000 years ago and they diverged every 1,000 years, we'd have current diversity in 11,000 years. So we've still got like 50,000 years worth of diversification to go. And we don't see nearly that else. But that's obviously very simplistic, but I think it's useful in helping us think about what questions we should be asking. So we were always talking about why is there so much diversity? Maybe we should be asking as well, how much diversity should we expect to see? And maybe there's less than we expect. And again, this doesn't really work because Australia has a similar time depth, the same time depth, but there's much fewer, comparatively fewer languages in Australia. So something different is happening between those two places. And the final hypothesis I mentioned is this multiple founding group hypothesis. This is the idea that people are just continuously coming into the New Guinea and each time coming with a new language. And that language is diversifying, that's why we have so many language families. So if you're making a basic guess based on how many language families there are, maybe you suspect there are 40 migrations or something. No one's proposed that, I'm just speculating. And so this hypothesis, you'd think would be amenable to genetic analysis, but the genetics of this doesn't really support it either way. It's not really for or against the multiple founding groups hypothesis. There's some simulation events that suggest this is a likely hypothesis that if we want to see current genetic diversity, that must have happened in some way. And there's some archaeological evidence, I think, although maybe the archaeologists will correct me. Yeah, so a lot of these, the basic summary is that a lot of these hypotheses are, fuzzy, I guess. One point on this one is that a lot of genetic diversity arose in New Guinea around 10,000 years ago, which would suggest that the time depth maybe isn't as important. But the problem that we ran into when we started investigating this is that a lot of the genetic sampling done in New Guinea is of the Trans-New Guinea family, which we also think started spreading around 10,000 years ago. So there's a lack of diversity in genetic sampling in answering these questions. So that's kind of where XE came in and was like, okay, how can we kind of broaden the diversity of this genetic sample while also looking at linguistic diversity? But first I'm talking about why genetics is going to be useful for this question. Obviously, genetics is passed on from parent to child. You get some genes from mum, some genes from your dad, and it's likely you'll also get a language from the parent. It's going to be a little bit different to genetics because your parents probably speak, in the case of New Guinea, they probably speak a lot of languages. The chances are the first language of your father is different to the first language of your mother, but maybe they both speak that language. So you will inherit some language that your parent speaks, but not necessarily every language, I guess. But let's just say that we're happy with this assumption of parent to child language transmission. If populations remain isolated for centuries and centuries, then we're going to see really strong correlations between genes, language, and then also geography. And if that was the case that we saw in the data, then we might start to think that these hypotheses have some merit. And there's some kind of isolation time depth, depending on how the relationship looks, evidence from the genetics on why linguistic diversification might occur. Important note is that New Guinea, in the areas that we look at, is pretty much all patrilocal. There's some exceptions in other areas of New Guinea where we don't have genetic samples or don't use genetic samples. And also we're going to be looking at allosomal markers, so sex-biased genetic markers. So mtDNA everyone gets from their mother, and then Y chromosome data sons get from their father. And so we're testing this idea that whether these kinship systems are creating durable channels for language transmission, which is something that Stephen J. Lansing and Murray Cox have written quite a lot about. So this is the extent of genetic sampling in New Guinea. You'll see it's pretty much all Papua New Guinea. We have some goal to get some genetic data from here, but we don't have any at the moment. And so there are blue crosses existing samples. A lot of it's collected by the Papua and Pass project, which is run out between the University of Toulouse and the University of Fiji. But ICTI has started collecting samples in southern New Guinea. So we have 92 samples that were collected in 2019, another 40 that are going to be sequenced soon. And so our sampling is trying to get across the linguistic diversity of this area, whereas the majority of these languages are trans-New Guinea. Well, not these ones. So this is the picture of southern New Guinea and where the language families lie, and where we're interested in. The border between Papua New Guinea and Indonesia is roughly here. But we've got linguistic samples from the Yam family, Victoria River, as well as a couple of these branches of TNG. And so in the future, we're going to examine this in more detail, but I haven't done it yet. So I'm going to talk about more broad scale stuff that I can leverage using the data that already exists. So this is, I've done a lot of phylogenetics in the past, so we went down the phylogenetic route. And this is the phylogenetic history of the mtDNA data, so the material you get from your mother. If you don't know what a phylogeny is, this is 0, so these are the people we sampled. Each little dot color, which are overlapping quite a lot, but it's because there's a lot of people. Each dot is a person, and the color is the language family they speak. Then the lines here are indicative of how closely related they are to each other. So, and because we know quite a lot about how mtDNA accumulates mutations, we can more or less time the amount of time. we can estimate the amount of time passed between any two individuals. And so we get a time depth of around 45,000 years, which is consistent with a lot of other genetic studies in the area. I should note this is just the N haplogroup. There's two major mtDNA haplogroups in New Guinea, N and M, but they tell pretty much the same story, and it was like quite a lot, so I'm just going to focus on that. And then we have another phylogeny for the non-recombinant Y samples, which is only about 70 people. That has much deeper time depth, but that is probably just a result of two haplogroups being entering into New Guinea rather than indicating any time depth for people in New Guinea. That makes sense. And one other thing I should mention is, because this is still relatively early results, we started out looking at language family affiliation, which is quite an odd category to study for this, I think, because language families don't really mean anything to people who live there. They don't mean anything to anyone really, except linguists, I guess. In terms of how you communicate with people, you either speak the same language or you don't. And so I have this kind of crude justification for using language families that if you're going to learn multiple languages, you're going to find it easier to learn languages that are more closely related, maybe. So it might be the case that we observe some kind of restriction of movement based on language families. But to head off any questions, I plan on doing it on languages and other categories later. But the question we're interested in is does language family affiliation constrain genetic diversity? And that's what this graph on the side is trying to tell us. So blue is the probability that you share a language family with someone in your genetic clade, which I'll explain in a second, and green is just random chance. So to conceptually explain how we calculate that probability at any point, you can, if you imagine a phylogeny like a children's mobile, and you like cut it across, you cut the strings all across at one point, you'll be left with lots of little mobiles, and you look at the people in each of those little phylogenies and you calculate what's the probability of two people sharing a language family. And you do that for every one of the little phylogenies, and then you take the average, that gives you the probability. And what's this is kind of showing that language family affiliation is a pretty good predictor of genetic diversity up until around 15,000 years ago, maybe 10,000 years ago, which is more or less when we think the transgenic expansion happened, which is the majority of the data. Just to explain the error bars, so this is the error in the phylogenetic structure. So I'm showing the average phylogeny here, but we have around 10,000 different ones that are all changed a little bit. And then the green is a combination of the range and as a result of just permuting, like randomly assigning the language families versus the phylogenetic diversity. But as I said, yeah, random chance is a pretty terrible hypothesis where you think that people will probably be closely related than chance when it comes to linguistic diversity. So then we ask a better alternative hypothesis, which is isolation by distance, which is the most basic diversification model of genetic diversity. The basic idea being the further people are away, the less chance you've reproduced them, I guess. But the problem with the isolation by distance, it's not necessarily a problem. The problem for us is that we don't really know what a good distance would be. So I just kind of picked three. If you look at 200 kilometers, it's almost always the case that you're close to 200 kilometers within. That was a terrible choice. New Guinea is, like, as we said, is not a very big place. So almost everyone was 200 kilometers from each other. When you get to 100 kilometers, the relationship is getting very overlapping a lot. So linguistic affiliation and geographic distance are pretty similar at predicting genetic diversity. But it's not until you get to the smaller distances, 50 kilometers, that you start to see linguistic diversity, linguistic affiliation being a better predictor than distance. And we have some anecdotal evidence that the marriage distances are commonly less than 50 kilometers. So getting to like a day, maybe 2 days walk away from the village that they're in. So we're putting more weight on the shorter distances at the moment. This is the same model but for the non-recombinant Y. It's a lot more jaggedy because we have way less people. So there's about 850 people in the previous one. Here we've only got 70. But we see a similar pattern. So green is not chance, it's distance. There's distance in language family affiliation. It's as a bit of a predictor as distance gets restricted. So we're taking this as like preliminary signs that language affiliation is constraining genetic diversity in some way. There's obviously proximal evidence of this, proximal evidence for this. I think it signals that there's this cultural constraint of genetic diversity, culturally mediated to isolation, I would call it. So it's not necessarily isolation in the way I described it in the hypothesis earlier, but there's some kind of cultural forces stopping people from interacting with each other. And I think that kind of fits well with what we know about New Guinea at the moment. New Guinea in the past, I guess, but from current literature. But this is my big caveat slide. There's heaps of caveats. So first of all, we're just looking at language family. It's quite a broad category, and as I said before, it's not that same. So we need to go down to the language level, which is what other people have done. The problem with going down to the language level in New Guinea is that there's just so many languages that most of the time we've only got one person sampled from that language. And so immediately the probability of them sharing a language goes to zero. So we're going to have to think about how we're going to calculate that. So we have plans to go down to the language, but there's some complications we need to work through to figure that out. We also should test on more specifics. So this is just on average, right? This is on average how likely people to share a language within a genetic type. But we might have reasons to believe why some particular language families have more or less steps than another. And can we parse that out using this approach? And finally, is that we're looking at just 2 genes here, right? So Y and mtDNA, but humans have loads and loads and loads of genes. So I'm working with Ray Tobler, who's a geneticist at ECTA. He's a population geneticist, so he's more interested in getting to more complex models of population history and whether we can intersect that with more complex models of language history as well. But this is kind of early day stuff, I guess. Think of like, oh, that is about 40 minutes, is it? My timer on here says 59 minutes, and I was like, this not. Yeah, so my takeaways, New Guinea is the center of global linguistic diversity, not only in terms of the number of languages, also has incredibly high disparity relative to the geographic size. But with the caveat that Austronesian is also showing huge diversity, and I'm not really sure why. And then we have this proximal genetic evidence that linguistic affiliation is structuring genetic diversity from around 10,000 years ago. Yeah, so my thank you slide. Thanks to Ray and Nick. Then Nicholas Brocato and Francois Xavier, who we work with at the University of Toulouse. Ashley Fowler, who I think is based here now, but he was at University of Melbourne, and Betty by UPNG has been talking with us about her ideas. So yeah, thank you. Yeah.
Just a couple of questions. The Austronesian diversity is surprising, but then when you consider how absorptive a lot of these Austronesian spatial communities are, and like to sort of capacity to absorb genes, using the other practices as well, then you'd expect, given the geographic range of Austronesian, that it's hooking into multiple forms of diversity from Madagascar and so Solomons. So that would explain that.
That's
diversity within Austronesian.
Yeah, that's kind of the same with what Nick was saying.
Yeah.
And when I was talking about like the internal structural diversity, it's just an indirect way of being, it could be that they're borrowing social organization ideas, and that's feeding onto kinship as well.
Yeah,
that's a similar idea. But yeah, this could be context stuff.
So the other questions about your distance, measures of distance in looking at language affiliation, and I just wonder if we can get more granular, because 50 is obviously much more informative and productive than 100 or 200.
Yeah, we can pick.
But can you do 25?
Yeah, we can pick any distance.
So that would be really interesting, because my strong suspicion is that you find 5 kilometers is actually a bit of a limit, 5 to 10. And so I wonder if you actually did, well, much more granular intervals from 50 to 5.
Yeah.
If you actually pick a clip, which would give you a much more sensitive indicator of maximum or distance.
What I actually want to change to in this calculation is time rather than distance. Travel. which I think would be more informative, but it's the same idea, right? Yeah, you get your flags, and then you'd be looking at things like, is a day, and then like, you just do it for like 3 minutes or something.
Yeah. And then you've got some spectacular anomalies, like the people that produced greenstone axes and islands, or axes and islands. John Burton's done work to show that they can suck dead women from incredible distances when they're producing this biograph item. And so particular centres become actually have much broader range than others.
Yeah,
That's very granular.
and it's similar to what I was talking about no writing down, because we're just doing on average across all language families, but you might expect, like you say, some particular language families, different patterns, which is something we look at, and it's going to be a function of how much genetic sampling we have from any particular area. But
why did you make these three time periods?
these distances,
Yeah,
for example,
I mean, but those are the only, you didn't like fiddle around with it, you got to find the story, you know what I mean? Like,
yeah, well, basically, I needed to get some results, and so I just checked 3 and
oh, you didn't check.
and then we talked about them, and they were like, oh, this is definitely wrong. And then I've just been like flat out since then, because it's all like pretty new, like running these results.
But the other one is interesting. It's like a perfect interaction where it goes blue on top, green on bottom, and then they're the same. Yeah. And then it's flip flopped in the next one.
Yeah, I mean, because the structure is heavily based on a phylogenetic structure, because they're both based on the phylogeny. Right? That's why they both have this kind of curve like this.
Yeah.
Because you have this process. If you go back to the empty DNA phylogeny, you can see you get like a burst of diversification here, and then another little burst here, and then it's kind of flat from there. So the curve is kind of based on the branching of the phylogeny. But yeah, I'm not sure that answers your question.
How did you pick what language? Like, everyone's very multilingual. Is this like you go?
Yeah, they're very multilingual, but they still have like a home language.
Oh.
Like this is the language of the village I was born in, but they might speak lots of languages.
Because it must be very patchwork. It's like how language they speak versus the ones that
Yeah, and that's because we had way more mtDNA data. That's another reason why I went with language family first. And we don't have much Y data, which is the pattern you're kind of suggesting, right? Is that you would expect higher probability in men and than you would in women.
Yeah, because they use leaving.
Yeah.
Yeah, exactly.
Yeah, exactly. And that's what you find in I forget the name of the islands, but in the island Southeast Asia with Stephen Lansing and Murray Cox stuff.
Oh.
No, it's
Sorry, it's East Timor area.
yeah, East Timor area.
Stephen Lansing doing genetics now, it's near cultural anthropology.
Yeah, but Murray Cox is a geneticist.
Speaking of that, one of your model maps, I think it was one showed surprisingly high diversity in Southeast Asia as well.
Yeah.
What is that mainland and island?
Sorry.
Is that mainland and island? So does that include Eastern Indonesia, for example, or is it mainly the mainland? Because there's multiple language families there.
Yeah. I'm not sure.
That's right.
So this was done with Simon Greenhill. He did the cutting up. I don't know exactly where the dimensions are for it. But also quite a lot of this. But I think the main thing is like the main thorn from this is there's been this assumption that typological diversity in language is indicative of time depth. And maybe that's not something we can assume that strongly.
But a question for Peter Bellwood online. So
Oh, yeah.
like,
He came to my last talk. Yeah,
would you like to come in here? Or would
Yeah.
you like to do the others?
I can do it. How do you do it?
Click on
Attendee.
these.
I can't do it. So yeah. So.
So you might hear you.
Okay, hang on. Give me a second to do that.
Okay, Peter, can you hear us?
Can you hear me?
Yes, we can. My computer is loud as it will go. Sorry, folks.
That's very good, because I can't hear the discussion. I heard the seminar very clearly and the speaker, but I can't hear what you've all been talking about since. But I've just got one question. I mean, what used to be called the Trans-New Guinea phylum, you're now calling Nuclear New Guinea. Is that correct? Did I understand that clearly?
Nuclear Trans-New Guinea.
Nuclear Trans-New Guinea. Okay. What do you think about? You're a linguist, right? Not yes. You're a linguist. So
I'm not really.
No, all right. You're a computational analyst, put it that way. But do you think that well, let's call it Trans-New Guinea for simplicity. Do you think it's a proper language family with an ancestor and a kind of, a history of origin and diversification through spread? And if so, do you think the spread was attached to any human population? I know there's no archaeology or ancient DNA that could ever throw light on this, at least not directly. But how do you still regard it as, I presume, the largest language family in New Guinea?
Yeah.
How do you think it originated, and do you have any ideas about its age? The coalescence age for the languages.
Well, if we were going from the graph, I just switched the computer.
Bring this over to you.
Okay, thank you. So if we're looking at like this graph here, right?
Yes.
Most of the individuals we have are Trans-New Guinea speakers.
Yeah.
So let's just pretend that they were all Trans-New Guinea speakers. Some distinction. Then you're getting to around like 10,000 years old.
Yeah.
I think that's kind of what the genetic stuff is pointing towards. It's not, I mean, I haven't specifically looked at that, but I think that's what it would be pointing towards, is this kind of 10,000 years.
I can't, in a graph like that, I can't really see that, I have to say. I know there are a lot more sort of little sort of splits at around 10,000 years ago. Yeah, maybe that means something, yes.
The function of the linguistic family location and the genetic structure, right?
Yeah.
So that's what the graph on the right is kind of like how those correlate with each other. And they don't really correlate past 10,000 years, but they do after. Well,
of course, 10,000 years does bring up this question, could it be related to the origins of agriculture in New Guinea? I'm not sure that was 10,000 years ago. It might have been 6 or 7. But still, I know it's very difficult to answer these questions. And I'm always surprised that in Australia, the Palmer-Nyungan languages are so, or appear to be, according to linguists, so closely related. I wonder how they spread. I'm not going to discuss that now, but I mean, that is another very interesting question, because the current DNA records, which of course are entirely on living people, not on dead skeletons, suggest that there have not been recent movements in Australia, at least not sufficient to cover the whole of the Palmer-Nyungan area. And that's still a bit of a mystery.
Had a paper come out last year, I think.
Yeah, it did.
Comparing the linguistic and phylogenetic histories, which is quite good.
Okay, all right, no, that's all. Thank you.
All good?
Yeah, that's fine. Yeah, thank you.
Thanks. Awesome. Is there any other questions for Sam in person or online?
I have one quick question. Yeah. It was about part of the background, but I didn't quite understand. So can you back that up? Like, I think might be one slide back there. More. These, no, the next one. So what's being measured on those two axes? Those are principal components, but do they have any?
No, they don't have any meaning. It's more about distance between points. So the algorithm is trying to show that, so the distance is really multi-dimensional, right? Like geographical distance, but then like 3 dimensions, 4 dimensions. So how can we most accurately map that distance onto a flat surface? So yeah, the axes don't really have any meaning other than helping me put plots in the right place.
And then the colors, like in some of them, the colored area is incorporating all the dots, and then the other ones. What's that mean?
Every graph has the entire distribution at the bottom. So all of these, this point here is still here. This point here is still here.
Oh, I see. Oh, okay. So it's the same dots.
It's the same dots underneath. I'll just show you where the language family is.
Yeah. Thank you.
Awesome. And there are no other questions online. So I will get everybody to join me in thanking Sam for his awesome talk. And I'll let everybody know that our next speaker is Associate Professor Karen Barb from the Department of Anatomy at Midwestern University, which is in Arizona. And she's going to be speaking of evolution and variation in Pleistocene Homo. And that's on the 19th of April at 1 p.m. So hope to see everybody there. And thank you again, Sam. That was really interesting. So, yeah, and thank you all for coming.